In the five kingdom classification system, the third kingdom which we study about is the fungi. Fungi includes the organisms which have some similarity with the plants and some other similarities with the animals. So they can be said as the link between the animals as well as the plants. Now, when the scientists started thinking about the fungi, they always thought of mushroom. And with this, the term mycology came. Mycology means the study of mushrooms, or in simple terms, in Greek, it was said that mycos means mushrooms, and logy means discourse, is to talk about or to study about them. These organisms, they are almost found in almost everywhere, but they grow the best in hot, humid kind of climatic condition where there is some moisture present in the atmosphere. They grow even well in the dark conditions. But not only this, these are the ideal condition for its growth, but other than these conditions, they can be found in the areas where even normal climatic condition is present. Coprophilous or symbiotic mode of nutrition can be found in some of the fungi. Coprophilous is a condition in which fungus is drawing its nutrition from dung. The dung can be a cow dung or any other feces. So just they will be growing in an area where feces is present, and that gives the term, or that is the meaning of the term coprophilous. Whereas some other bacteria like fungi, they will be depending on some bacteria or some other plant or organism which will be making the food, and these fungi, they will be providing some nutrition to them. Symbiotic relationship just means having a partnership with some other organism, and the partnership is helpful for both of them. One of the organism, which is this fungus, is incapable of making its own food, so it will be getting its food, and since it is capable of absorbing nutrients and water, it can supply it to any organism who can prepare the food with the help of mineral as well as water. They are capable of absorption of water and other substances from outside. If we see the structure of just one of the fungal cell, you can see this structure. This is a elaborated structure of a fungal cell. Mitochondria, the function is the same, that it will provide or it will form the ATP. Vacuoles, they store water and some amount of storage product, and they provide turgidity to the cell. If the cell is much more turgid, it will be capable of absorbing nutrients, water and minerals at a very rapid speed as compared to flaccid condition of vacuole. Every cell is completely visible, or it is well expressed when there is turgidity in case of vacuoles. Function is the same, it will store water. Glycogen is the storage food of the fungi, and since in human beings also it is glycogen which is the storage food product, so this created the confusion of fungi being belonging to either animals category or to plant category. So this is a confusion because of which the kingdom fungi was formed. Next is about the dictyosomes or the endoplasmic reticulum. The function is the same. It forms the proteins and it supplies the proteins to the different organelles where it is required. Presence of ribosome is there, and these ribosomes, they are helpful in protein synthesis. Endoplasmic reticulum is not only forming the proteins, it is also forming the enzymes and every other substance which can be formed and transmitted or packaged and delivered to the area of its work. Nuclear envelope is present in these fungal cell, and as because nuclear envelope is there, this gives the cell a name, that is eukaryotic condition. So eukaryotic condition is maintained in case of fungus because of presence of nuclear envelope. Nucleolus contains the protein synthesizing property. Nucleus has the genome of the fungal cell. Plasma lemma is the outer extension or the outer plasma membrane kind of outer covering which separates the inside or intracellular content of the cell of fungi from the external or extracellular content of the fungus. Cell wall is made up of chitin, and Cell wall has presence of lomasomes. Lomasomes are certain structures which have or these structures are as such that they serve the separation, or they secrete some crystals or granules which separate the cell wall and the plasma membrane of the fungus. If we see the characteristics or general characteristics of the fungi, first thing is that they do not have presence of chlorophyll and they are heterotrophic. It means they are either parasitic or saprophytic. Parasitic means they are drawing the nutrition from some other host cell and harming the host cell, whereas when we say that they are saprotropic, it may or saprophytic, it means they are drawing the nutrition from the dead and decaying plants and animals. The entire body of the fungus is made up of hypha. So think about straws. I have given you 10 or 15 straws. Hold all of them together and think that all of the straw, one of the straw I'm just trying to make, it is of this type. So hundreds of straw are there with you and you have arranged all of them together and made a bundle out of it. This is the way a hypha looks like. This entire structure is known as mycelium, and if I say or if I just take out one of the structure, we call it as hypha. So I think it is quite clear to you all, the entire body of mushroom is made up of, or not only mushroom, any fungus is made up of lot of hypha arranged together in a bundle kind of structure, and that entire bundle is known as mycelium. Mycelium is working, or it is looking like a entire body. Next is the storage food material. We have already discussed that it is the glycogen in case of fungi, and the cell wall is made up of cellulose and chitin. You can add chitin over there. Chitin is proteinaceous in nature, and glucan is the main or n glucan, or we can call it as n glucosamine, which is the main polysaccharide which helps in formation of the outer cell wall of the fungus. The nuclei is present in case of fungi, and nuclei is not only present, it is even the nuclear membrane or nuclear membrane containing nucleus is there, which makes it a eukaryotic organism. The As the cell is progresses itself, the kind of sexuality it is about to express, it gradually decreases. So although the sexual reproduction is present in them, but it is not well expressed. And we are already aware that these fungi, they are saprophytic, and because of that they decompose all the organic matter which is as such dispersed in the environment, and because of that they are known as the decomposers of the environment, or even the mineralizers, because every substance or organic substance is containing some minerals within it, some carbohydrate protein subunits within it. So when it is completely decomposed, all the minerals and uh, proteins, they will be mixed with the soil, and that's why they gain this name as decomposers. This is a structure by which two of the hypha, they are connected with each other. I said that each of the straws, they are known as hypha, and 10 or 15 straws together or hundreds of straws together, they form the mycelium. Now these suppose hypha 1 and hypha 2, both of them, they are connected to each other by somehow or some formation by which they are connected. That formation is either a micropore condition. Micropore condition means two cells will be there, or three four cells will be formed, which will be looking like a chain, and that with that cytoplasmic bridge, two cells will be combined or will be attached with one another. You can see the condition in the first diagram A, that small hexagonal structures are there which are connecting two septate or two different hypha together. Second condition is a simple pore structure where two septum just come very close to each other, and they are so close that they can exchange substances from one cell to another. Or you can think that cellular constriction is formed with the formation of this simple pore. Sometimes the pore region is as such that it has presence of some granules within it. or within the center of two septum, and that is known, that also helps in differentiating two cells. Doliopore is found in the highest organisms or in the most developed fungi. The function is again the same. It is centrally located, large jar shaped or barrel shaped structure which is quite swollen, and the cap thickenings which are present at the corners, they are known as parenthiosomes. These structures which are present at the corners, they are known as parenthiosomes. This is M and E, parenthosome. How these structures are formed? They are formed by deposition of Worlin bodies. I will just spell it. It's Wornin, Wornin bodies, and these Wornin bodies are some crystalline granules which are just secreted by the both the septum, and they form these entire doliopore, the region where it is set. These all structures, or this entire structure, is formed because of secretion of those granules by septum 1 and septum 2, and as a result both the hypha, they join with each other. If we think about the septum, we should think about the all the types of fungi which are present. Some of the fungi are unicellular like the yeast or Saccharomyces. So in them there is presence of pseudomycelium. Mycelium is the entire structure of the fungus. So here, if you see this structure, a budding structure is formed, and since it is a budding structure, it is not a mycelium or a hypha, like not the attachment of hypha is there. That's why we call it as pseudomycelium. Pseudo means false, mycelium means false hyphal arrangement is there in case of yeast. Aseptate condition is a condition in which the entire cell is present, or hypha is present, but the hypha is not differentiated. Here you can see that there is no differentiation of cells in case of C structure, and that's why it is known as aseptate condition of a fungus. This is seen in lower fungus or less developed fungus. Monokaryotic septate condition means there will be presence of cells or disc shaped cells differentiating two cells from one to another, and each of the cell will be having presence of nucleus within it. So you can see the presence of nucleus in each of the cells present, and it is known as monokaryotic. Dikaryotic condition is present when one of the cell will contain two caryon, means two nucleus. Multinucleated condition means one of the cell will contain many nucleus within it, and all these structures, they are septate. Now when we see the next structure, it is the same septate and non septate. I'm saying it is aseptate condition, but in a elaborated way or just magnified view of it is shown. You can see the disc separating cell 1 with cell 2. Both the cells, blue color nuclei are the cells, are the nuclei, whereas the disc shaped structure is the maybe a micropore, or it can be just a small pore, or a doliopore which is separating two cells, and this disc is known as a septum. So you should remember it, and when this septum is present, it is fungus is known as a septate fungus, or the hypha is known as septate hypha, whereas when it is absent, it is known as coinocytic hypha or aseptate hypha. Fungal structures and their growth. We have an idea how the mycelium looks like, and it is arranged with the help of several hypha, or it is formed because of arrangement of several hypha together. Each hypha has presence of some constrictions of cells within it, which is formed with the help of either a doliopore or with the help of some small pores with the formation, and that is known as septum. Holocarpic condition is uh, known as a situation when the entire fruiting body of, or the entire plant, entire hypha or mycelium starts becoming a reproductive structure. Whereas eucarpic condition, in the eucarpic condition, a small portion of the mycelium takes part in reproduction. septate and aseptate condition we already have an idea about. You can see that this entire structure of a mushroom, this entire structure is known as a mycelium. So several hypha, they all entangle amongst itself and then form the mycelium. Whereas if we think about just one of the hypha and separate it, it may have presence of this pore. This may be this, see, it is dumbbell shaped, the way we were talking about in doliopore. So it is a doliopore septum which is present, because you, if you just look into this structure quite carefully, it is multinucleate, sorry, dikaryotic doliopore septate condition present in this structure of mushroom. And you can just correlate with it. The septa, these all blue, these orange structures, they are the septum, and hypha, and they are separated by doliopore septa. Each of the hypha, like one of the hypha is cut from the upper part, and then after it is shown, and it is showing the septate condition. There are a few terms which we should discuss. Chitin is the outermost covering of cell wall in case of fungi. Unicisternal is a condition in which there is presence of endoplasmic reticulum as well as Golgi apparatus, which helps in transportation of enzymes and proteins in case of fungi. And this is a characteristic feature of only fungi. You do not see the presence of unicisternal uh, Golgi apparatus and endoplasmic reticulum together like a dictyosome in any other organism other than this fungi. So you should remember it. Chitosomes are, if we think about a hyphal structure, suppose this is a hypha which is formed, and at the end there will be presence of cell wall. Think that cell wall is present at this end, and the function of this chitosome is to supply chitin to this end, and this chitin will be helping in formation of the outer wall or cell wall. So that is chitosome's function. In case of fungi, proper centrosome is not present, and in spite of it we see the presence of spindle pole bodies. They secrete just tubulins, and those tubulin help in separation of cells or cell division. Doliopore we have already seen, it is present in the mushrooms, and it has extensions which have lomasomes present. Not the lomasomes present. They have the presence of certain granules which are secreted, which form the entire structure by both the septum. Plectin chyma is, we have seen that interwoven hypha are present in case of most of the fungus. Now, plectin chyma is when all these hypha, they are interwoven amongst each other. So when they are highly woven with each other, we say that it is in plectin chyma condition. Prosenchyma is when, is just the elaboration of this should be n. Prosenchyma is a condition when the hypha, they are loosely woven with each other, whereas pseudoparenchyma is a condition when the hypha is compact mass and it looks like a parenchyma like tissue. Although they are closely packed and interwoven, but still they have their own individuality maintained because of the isodiametric structure they possess. So if I want to show you all the structure of prosenchyma and pseudoparenchyma, it is somewhat like this. So you can see the isodiametric form. This isodiametric form is the pseudoparenchyma. So I will just mark it. This is pseudoparenchyma, and prosenchyma is this one. Plectinchyma, it means that both this interwoven structure or interwoven nature of hypha will be maintained, but are they arranged in a pseudoparenchyma arrangement or in prosenchyma arrangement, that is to be determined on the nature of the fungus. So this is how the fungus, they all have some of the characteristics, and we can study about them in by understanding these few things.